helicase is an enzyme that separates the two parent strands. It begins during initiation, and it's how we get the replication fork. Without it, we wouldn't have the two DNA strands could not be separated. So SSBs are proteins that are binded to the separated DNA strands, and it helps keep them separated because the bases want to get back together through hydrogen bonding, so it keeps them separated and stabilized. Gyrase is an enzyme that happens before, or that appears before the replication fork. It relieves the supercoil, the tension from the supercoil that DNA has, and makes the separation easier. RNA Primase helps with replication by creating RNA primers, which synthesizes complementary RNA, and it helps initiate DNA polymerase 3 and get it going. DNA polymerase 3 is an enzyme that actually does most of the replication. It attaches to this parent strand up here, and it moves in this direction, making synthesizing new DNA. Now, before we get to replication itself, let's clarify the direction of replication. This is 3', 5', so 3', 5'. The RNA primers are 5', 3', so 5', 3'. DNA polymerase 3 moves from 3' to 5'. However, the replication itself goes from 5' to 3'. And the origin of replication is self-explanatory. It's where replication begins, and it begins in this area, the 5'. Now, how does DNA polymerase 3 move and synthesize new DNA in the first place? These nucleotides, they consist of 3 phosphate groups, and they're attached by phosphodiester bonds. By breaking these phosphodiester bonds, it releases energy required for DNA polymerase 3 to use and need to continue to move and synthesize new DNA. Now, let's label this replication fork. This is called the leading strand, the top strand, and the bottom strand is called the lagging strand. Now, why is the top strand called the leading strand? Well, it's called the leading strand because the DNA polymerase 3 can continue to synthesize new DNA in this direction in a continuous motion, and as the helicase unwinds. However, as the helicase unwinds, it exposes bases down here that the DNA polymerase 3 cannot reach, so it can't synthesize new DNA in the lagging strand. So, in turn, it's lagging in terms of new DNA being made compared to the leading strand. Now, these Okazaki fragments creates these primers down here, and it's only in the lagging strand. However, to note that they are discontinuous, so it's 5', 3', 5', 3', 5', 3'. It creates new DNA, but it's discontinuous. Now, these DNA ligase is an enzyme that connects these Okazaki fragments together. It's really important because it makes this entire new DNA in the lagging strand continuous. So this DNA ligase right here, this is an Okazaki fragment, this is an Okazaki fragment. It comes in and it binds these two together, making them continuous.